Yeah, I'm sorry, there was a problem. This is a serious issue. I mean, can you see the board? Can you hear
Yes,
me?
sir.
Yes, sir.
Yes,
Yes,
So
sir.
let me sort of talk the way I want to talk. See, the type 1 pathway, as I said, this is the E. coli cell, and let us call it the inner cell, and this is the periplasmic membrane. And the proteins that sit across, they actually sit in Secchi, they sit across the inner membrane, but in Hyle and all, they sit like this, as I said, and they actually create a channel for the protein to go out straight away. Similarly, there are many proteins which actually, for example, pillin genes, pillin genes also have this ability to push this set outside, because that is how the pili is formed. It is attached to the outer membrane, but they actually grow from here. So there's something like the surface display that people talk about. The surface display genes, they are sitting on the external surface of the E. coli has a lot of signaling peptides and signaling molecules sitting on the surface. Many of these are proteins. How do they manage to sit on the surface? This whole problem of surface display, because they are pushed out. Similarly, viruses also tend to migrate to the surface, and the genes which are used to transport them or translocate them to the surface, the same genes can be used possibly to produce your recombinant proteins. Because after all, the promoter, the signal sequence, and then the gene. And it is the signal sequence, this part, which is actually the important part. And the signal sequence can actually, with help from the E. coli cellular machinery, actually ensure that proteins that are made here are sent out here. So that is the whole idea. But really, what you people are focusing on is a different problem, and what I was asking you was a different problem. You are focusing on the properties of the gene and its signal and its location to ensure that it is produced. This gene product goes out. The gene product goes out. But really, the question that I was asking was slightly different. What kind of cellular modifications do I create that creates holes in the extracellular membrane, so that once my protein has got translocated into this point, then it has a hole which allows it to come out? So the real question here is, can you think of genetic modifications in the extracellular membrane, not the intracellular membrane, the extracellular membrane, and the genetic modifications which will cause such extracellular membrane changes, such that proteins which are produced in the periplasm are actually secreted out? So really, and the answer to that is we do not know. We do not know, because obviously this E. coli cell does allow some proteins to reach out, but only very specific proteins. You want a much larger class of proteins, including the product of your interest, to come out. So the real question is, what host modifications do you do? Not changes in the gene itself. That is part of the story, but most changes in the gene, as I told you a long time ago, is usually with respect to signal peptides, and they push the protein to the periplasm. What changes in the host that you will do, that, so now it is not a question of genetic manipulations, but host modifications, actual genomic modifications in the host that will allow you to create novel hosts, the genomic modifications that will allow you to create model hosts that will allow the protein to get outside. Now, for that, the standard method is, if you do not know the answer, create a pool of say genomically modified hosts. How do you create such a pool? It's damn easy. You can always create a pool of genomically modified hosts by random mutagenesis. For example, genomically modified cells by, let us say, random mutagenesis. Random mutagenesis will create random mutagenesis can be done by simply taking cells, and I am sure you are aware of these things in microbiology. You just use UV, expose the cells to ultraviolet radiation, kill off 99% cells. That kills off 99% cells. The 1% which survives will almost all be mutated. It is not a big deal, because you have 10⁸ cells, 10⁸ cells. in 10 ml, let us say 10 of 8 cells, and you expose it to UV such that only 10 of 7 cells survive. And 9 into 10 of 7 cells are dead. 10 of 7 cells survive, and now you have a situation where these 10 of 7 surviving cells are all mutated. Now, if there is 1 in a million chance of getting a good clone, which is modified, random modification, so 1 in a million is a good hit, then out of these 10 cells would have secretory capabilities. But there is obviously a minor, not a minor, a very serious problem with this strategy. How do you find out which 10 cells out of 10 of 7 cells? You tell students to sort of screen each and every cell and see whether it is secreting? Imagine 10 million cells.
Secreting can be done. We can use some reagent which gives some kind of color to that protein, and in that way we can screen.
So one of the standard methods has been people using color-based screening, which if not survival, at least in a single plate you can put about 100 colonies. So for a million cells or 10 million cells, you will only have to use 10 million cells, मतलब 1 करोड़, तो आपको 1 लाख प्लेट चाहिए होगा, 1 lakh plates, है ना? And 1 lakh प्लेट बनाना थोड़ा इरिटेटिंग काम है. 1 lakh प्लेट 100 कॉलोनी ईच, और फिर उन 1 लाख प्लेट में आपको देखना पड़ेगा कि किस वाले में लाल कलर आ गया है. ठीक है ना? It is a tedious process, but there are papers in as high good journals as Nature where people have looked for higher color production. But the problem with color is that color can give a good indication of production and increase in production. But color is given around the cell. So you do not know whether the color is because of the protein production inside the cell or outside, which really means that you have to actually then see the color outside. That means you have to see a zone, a color zone outside, not just a color. You add something in the media which interacts with the protein, produces a color, and then try to see which of these cells has the largest zone. The one which has the largest zone has the maximum extracellular production. But as you realize that this is a slightly problematic thing, because you'll have to make about 1 lakh plates, and out of these you may get 10 cells which are good color producers on the outside. Are you getting this argument?
Yes, sir. About the protein is outside or inside. In that, for that we can use GFP. GFP will not automatically go inside the cell. We will design a GFP which will bind to our specific protein which we want to screen, and that GFP will not go inside the cell, and we can know that it is outside. The protein is outside.
मैं समझा नहीं, फिर से बताइए. GFP लेंगे आप. Yes, please go ahead. Now, GFP is green fluorescence protein, and it is a very popular marker used by molecular biologists. Yes. And GFP is what gives fluorescence. It gives. So, what will you do with GFP?
GFP, I will design that. I will make a chimeric protein using GFP and a marker that binds to our.
Yeah, what we do is we connect the GFP along with the gene of interest. So instead of monitoring the gene, which is difficult, and this is a very standard strategy of molecular biologists, you're right, where the gene is bound with the GFP, and so the fluorescence, which is very easy to see, is often visible in is an indirect proof that this gene is also being produced largely, because the product that you make is actually GFP combined with your desired protein. And usually we put a linker in the middle, but I will not talk about all those details. We put a linker, etc., a linker which allows the GFP to fold independently, this thing to fold independently. There's a small linker here, and the GFP gives fluorescence. This GFP part will give fluorescence, and so you can locate very quickly what is the expression level of the gene of interest by looking at the fluorescence in the cell. But how do you guarantee that the protein is being exported outside? That is my question.
We will use a GFP outside, not inside.
You will use a GFP outside. ये कहाँ से GFP outside कहाँ से लाएंगे आप? कैसे? What do you mean by that?
Like I have GFP proteins already produced, and that protein I will use for.
You will put GFP on the plate.
Yes, sir.
If you put GFP on the plate, the cells will eat it up because they consume all things, amino acids, proteins, yeast extract, everything. जब वो सेल ग्रो करेगा तो GFP को खा जाएगा. और वो GFP अगर आप प्लेट पे डालेंगे तो पूरा प्लेट फ्लोरेसेंस. हाँ बताइए.
It normally doesn't go automatically inside the cell, and that's why
it does not go inside because proteases are secreted by cell. Proteases are secreted, and these proteases will consume all the they are basically meant to break down. Slow protease releases takes place, very slow release of proteases, and these proteases tend to break down everything in the media and make it fit for consumption. Okay, so they will slowly degrade GFP and eat it up. But let us assume, for argument's sake, that it is not able to consume the GFP. What will this presence of GFP on the plate outside do for you? What is the help that you want to do with it?
So the entire plate would be fluorescent. So
yes, the whole entire plate will start fluorescing, and slowly as cells grow, this fluorescence will decline because cells are growing. They will be consuming a little bit of GFP. Let us say that not all the GFP is consumed. The whole plate will show one large fluorescence. इससे क्या फायदा होगा रत्नेश जी? प्रूव कैसे होगा कि आपका प्रोटीन बाहर आ रहा है?
Okay sir, then maybe
हाँ, तो सवाल है ना कि प्रॉब्लम. हाँ, so now let me go back and give you some background of how people have addressed similar problems, and that might give you a better idea, because since we are discussing this, so let me sort of give you let us get today half the class is on microbiology, but only half, please remember, which means 10-15 minutes. We need to start engineering quickly. We don't want to waste time, but yes, because this is a creative problem. How have microbiologists done this job? As I said, the simplest method for detecting enzymes, whether amylase, protease, etc., is what I just now talked about. Enzymes, etc., is that you take the plate and in the plate put the substrate for the enzyme that you are trying to produce, add the substrate, okay, and when the cell grows here, and it interacts with the substrate because it consumes the substrate or produces the product, you will see some kind of zone around the cell where the substrate has got converted to product. So this zone represents product and this zone represents a substrate. So the cell which has the largest zone has the largest amount of, remember, it is not proof of enzyme being secreted. It is simply proof that substrate has gone inside the cell and product has come out. And the largest zone is possibly the one which produces the largest amount of enzyme. Okay, and this is the way that you select for amylase by putting starch inside and looking for clearance zone. And you look at protease where you put casein hydrolyzate, that is skim milk inside, and see for clearance zone. Starch, you add iodine. You look for lipase, or you look for a large number of genes. All these ases means they would be degrading something or the other, and you put oil and look for clearance zones and so on and so forth. Okay, this used to be the standard strategy, and that is what they teach in microbiology in all classes. So therefore, if you visit all the whole country, you will meet professors in the country who will say that we have the best amylase producer, the best protease producer, the best lipase producer. Unfortunately, nobody listens to them because it is unlikely that they are telling the truth. Because robotic screenings, as I said, really you need to screen 1 lakh plates. And these are done by robotic screens now abroad to really isolate best producers. Because, you know, once you have got this best producer, then again you do the same thing, mutate and see whether you can get an even larger clearance zone. And if the mutation produces a larger clearance zone, somehow this mutation has helped in increasing productivity. One does not know how. One can do reverse engineering to find out what is it that the mutation did that increased the productivity by analyzing the mutation, which is a tedious job, but nowadays doable by some at the level of, you know, gene sequencing. The whole genome can be sequenced, and then you try to understand what mutation was helpful, and then you discover that there are 20 mutations, and then which of those 20 was actually the useful part is a very tricky and hard job done by R&D people in the industry. It is a very hard business. Yeah. Yes.
Reverse engineering.
Oh, reverse engineering. Reverse engineering is a very simple term. I mean, if I know how the result was, I mean, first I do random. The reverse engineering involves first doing some random changes. Random changes followed in biology by a good screening protocol. Good screening protocol identifying the desired phenotype, and this group screening identifies the best phenotype, the best or desired phenotype. Okay, so and then you take this phenotype and then you analyze its genome, analyze its genome, and then by the analysis of the genome you identify why it is best, and obviously there are many factors in this genome which are making it. Only why it is best is really saying which of the changes of this, and because you have now let us say you have only 10 changes, which of these changes led to this best phenotype, led to this, and then only that particular change you add on to the actual bacteria. So this is reverse engineering. You are not doing engineering, you are not doing design, because your knowledge is inadequate. But since you do have a best phenotype, you reverse engineer it. So that is what the Japanese did with the Americans in the 1940s and 50s. They bought their cars, they bought their TVs, and they took it apart. They knew they did not know how a TV works. They did not know how a good car is designed. They bought these cars, took them to Japan, and then made Toyota, right? From the Mercedes or from the... So because they had a good car, they could take it apart, and then they had a very big advantage, even though otherwise their knowledge was poor, that these are the things that go into a good car. And then they could make a Toyota much faster. They did not have to do basic research the way Mercedes or other things did 100 years of research. Ford did 100 years of research before coming up with a good car. They could do that shortcut because they had a good car already with them. So they could take it apart. So you do random changes because you really do not understand how the bacteria behaves in order to provide you the best phenotype. You then do a screen which identifies the best phenotype, and then you analyze the good car, the good bacteria, and say, hold on, this is the reason why it was the best. And so now this particular change can be specifically incorporated rather than by random changes into a bacteria. So you have reverse engineered the phenotype. So reverse engineering is a very useful method for generating much better phenotype. So it actually follows the microbiologist's argument कि random changes करके भाई साहब best identify कर लो, and then it follows one step further by analyzing the genome, looking at the changes that led to this advantage, and then reverse engineer it or introduce it back. So this is how you reverse engineer the good properties, because a random mutation, even if it is the best phenotype, would carry all kinds of unnecessary stuff as well. You see, you understand that the best phenotype would also carry a lot of unnecessary baggage of changes. You don't want them. Maybe there are 10 changes out of which 2 are useful in this best phenotype. And these 10 changes are random, but 2 of them were useful. So just pick up those 2 and introduce it. Don't make all the 10 changes, because that has happened randomly, because when you were doing those changes, they also got incorporated. You get my argument? Yes, all of you?
Yes.
Yes. Yes. So now let us go ahead and let us sort of explain what we do. Now, this is the simplest of screens. Now, how do you make clever screens? And I really, because we are running out of time, I'll just talk of, and there are called, I mean, this is all creativity. How do you make, I mean, for example, how do you make a screen for antibiotic production? How do you make a screen for antibiotic production? You make a screen for antibiotic production by this very clever method, or what we call as bioassay. The bioassay involves the following. Now, I have a plate, on which I will grow my, I'll grow my microbes. Now, which of these is producing an antibiotic? Now, the antibiotic assay is damn difficult. So what do? I take a mixture of cells which are antibiotic sensitive, and highly sensitive to the particular antibiotic that I'm interested in. And I take this, grow these cells, add my agar, and then overlay this cells with my cells. Now this is fully these cells. I mean, this is all. I mean, this blue thing contains millions of cells, and they will all start growing and form a layer of growth. So on the top here, you will see a layer of a complete growth of this blue colored bacteria. Everything will be growing in blue. But this red has produced some antibiotic which kills this cells. So here you will see that there is no growth. And here you will see there is no growth. And here you will see there is no growth. So this red one has produced the largest amount of antibiotic which has killed the blue bacteria. And so you have a screen where you have, this is a bioassay screen where the blue colored bacteria is now dead because of the bacteria at the bottom. And then you sort of dig this one out. Digging it out is not difficult. It will be contaminated with blue, but doesn't really matter because when you grow the red, the blue will die. So you can grow this and the red will produce antibiotic to kill the blue, and then you have isolated the red colored bacteria which is producing the largest amount of antibiotic. And now you can again do the same thing. You can do the mutation on the red one and see whether you can get an even larger clearance zone. And the larger the clearance zone, the higher is the antibiotic production. And that is how people have done it, because antibiotic production is a secondary metabolite. The feedback controls are so enormous that it's impossible to design such bacteria, and you can only do reverse engineering after finding out the best strains. And that is how antibiotic production has improved over the years. And that is why all of us are alive. Because, you know, I don't know whether you've seen that graph, the production of antibiotics in the 1940s and the titers. And the titer was, if I were to say, it was at 10 units of production per liter in the 1940s. By the 1990s, in 50 years of this hard work of unknown people, you know how much the titers were? How much? 10 times increase, 100 times increase, kitna increase kiya hai? It was increased to 10⁵. So there has been an increase of 1,000-fold. Actually, sorry, I made a mistake. There's an increase of 10,000-fold over 50 years. The antibiotic titers were increased 10,000 times by this methodology by unknown scientists working in R&D labs for their whole lives. 10,000-fold. Of course, out of these, about 50-fold increase was contributed by fermentation design because the fermenter was run much better. But the remaining 200-fold increase was achieved by simply mutation. This was the contribution of fermentation by 50-fold increase. So from 10 units, the production became 10⁶ units. And that is why today you can go to the market and buy an ampicillin tablet for $5. When it was launched, only the absolute super rich could buy this. Just like it is the case with proteins like monoclonal antibodies. Nowadays, monoclonal antibody treatments in this country still cost 5 lakhs, 10 lakhs. So the poor can't afford it. Today, anybody can buy an antibiotic. So this is the kind of improvement that one is talking about. So these are the secondary speeds. Now, I will quickly, okay, I think we will lose this lecture, but it doesn't really matter. We don't have to bother too much. I will now quickly say, now what has happened because of overuse of antibiotics? There is antibiotic resistance in bacteria.
Yes, sir.
Now there is antibiotic resistance, which is especially in our country because of random and massive overuse of antibiotics. Antibiotic resistance has emerged. How does antibiotic resistance work? I have told you. It works by the fact that the cell actually gains a plasmid, usually a plasmid, sometimes in the genome, but usually by plasmids. And these plasmids carry genes. These genes, for example, for the case of ampicillin, I will talk of ampicillin. This gene codes for an enzyme called beta-lactamase. And as I told you, ampicillin is a penicillin derivative which contains a beta-lactam ring. And this beta-lactam ring is degraded. This beta-lactam ring is attacked by this beta-lactamase and degraded, so the ampicillin gets degraded. And so this cell can survive in the presence, it is ampicillin resistant. The cell becomes ampicillin resistant. Now, what do? We start discovering other new antibiotics against which it doesn't have the gene. Now, discovering new antibiotics is a very, very hard job. Extremely difficult business because there are not so many antibiotics. Far better is to discover something. Yes, now tell me, what do we discover? What is a better idea? Discover something, some chemical or enzyme or whatever. Let us say chemical/enzyme. I'm not interested in whether it's a chemical or enzyme. I'm interested in or enzyme or product, whatever, which converts this ampicillin resistance to ampicillin sensitive. How will you discover that? That's a better idea, because now suddenly if you have discovered that chemical, this chemical can go into the tablet. Now my tablet that I sell in the market will contain ampicillin plus this chemical. What will it do? It will ensure that it works on such that ampicillin works on this. How will it ensure that? Because this chemical here. Yes.
So how do you ensure that it does not work on like the host cells?
How do you ensure that it does not work on the
host cells? Like what if the chemical makes the host cells ampicillin sensitive?
This cell sensitive or the production cell? When you say host, it means production cell.
No, sir. Like if you talk about tablets, so like if a human takes a tablet and if the chemical makes like our cells ampicillin sensitive.
Our cells, we are not attacked by antibiotics. You know, let us. वो आप थोड़ा अभी हम लोग सिर्फ बैक्टीरिया और प्रोकैरियोट्स की बात कर रहे हैं, ठीक है ना? हमारा सिस्टम बहुत कॉम्प्लिकेटेड है. I mean, even yeasts are not attacked by antibiotics. Antibiotic, ये सब चीजें हम लोग we have all kinds of very complex structures where I mean, with it's a more. Let us not get into basic biology. You know, when you take the tablet, or it may also be injected, the blood, how does it react, and all these things. I mean, how does it get targeted to the particular place? That is a lot of basic mammalian biology. I don't want to get into. Let us talk of simple things like bacteria. I am asking a much simpler question that. An ampicillin-resistant cell generates its resistance against. How does ampicillin reach the human body to the cells, to the bacteria? Have you thought about that? First, answer the more simple question. How do you ensure after ampicillin, आप तो खाते हैं उसको, वो पेट में जाता है, उसके बाद कहाँ जाता है?
Sir, blood circulation system.
Blood circulation से ही जाता है, but there are all kinds of other mechanisms. Blood से फिर वो actual infection को कैसे target करता है, वो एक बहुत कठिन सवाल है ना? So let us not get into that. But you know that it is slightly complicated. So now I am going to talk only of the actual prokaryote. This is ampicillin resistant because it is producing beta-lactamase. So this chemical, it becomes very obvious. This chemical should be a beta-lactamase inhibitor. Understand? This chemical should be a beta-lactamase inhibitor. Because then when you have the ampicillin along with this chemical, then ampicillin will kill all cells other than those which are ampicillin resistant. And these cells survive and create trouble for you. But now, along with ampicillin, you are having this chemical, and this is a beta-lactamase inhibitor. Now, even this ampicillin resistant cell, it will produce beta-lactamase to counter the effect of ampicillin, but now because of the beta-lactamase inhibitor that will attack this beta-lactamase, it will no longer be effective. So even ampicillin resistant cells will die. Now this is not a vague statement of theory. आप लोग टैबलेट खरीदते हैं, ये beta-lactamase inhibitor क्या है? भाई कभी तो दवाई खरीदा होगा आप लोगों ने एंटीबायोटिक आजकल?
Yes sir.
Hello. हाँ? हाँ तो भाई आप में से in this class somebody should tell me what is this beta-lactamase inhibitor that is present in nowadays all antibiotic formulations sold in our country, almost all. कभी तो दवाई का स्ट्रिप देखते होंगे आप लोग, आजकल बिकता है नोवॉक्स प्लस समथिंग, याद आ रहा है? No, it begins with a C. Have you heard the term clovinic acid? टैबलेट में लिखा रहता है with plus clovinic acid.
Yes.
तो भाई क्यों लिखा रहता है भाई? आप लोग क्या मतलब आप लोग कभी तो पढ़ा करिए ना, क्यों रहता है क्लोविनिक एसिड? आजकल हर एंटीबायोटिक जो है क्लोविनिक एसिड, नॉट हर एंटीबायोटिक, मोस्ट एंटीबायोटिक्स, थोड़े से महंगे एंटीबायोटिक्स आर ऑलवेज सोल्ड एंटीबायोटिक प्लस क्लोविनिक एसिड. Why? Because of this. The clovinic acid is the beta-lactamase inhibitor. Now, beta-lactamase inhibitor ensures that the antibiotic will work not only against ampicillin-sensitive cells, which it will not naturally work, but also this combination will work against ampicillin-resistant cells. That is the whole idea, right? Now, my question is, how was clovulanic acid discovered? How do you discover a cell or a bacteria which will produce clovulanic acid, because that is an easy discovery to make rather than finding out a fungus which will produce another new novel antibiotic. See, because the clovulanic acid, you did not know at that time that clovulanic acid was the answer, but you found it out by reverse engineering. Okay, so how did they decide which bacteria will produce clovulanic acid? How do you design a screen to production of clovulanic acid? Bataiye. So what do? Now, let us have some, I think I want to be guided through with this strategy. So I now have the same strategy. What do? I have a plate in which I am screening bacteria. I am screening bacteria which are growing here for the potential possibility of producing clovulanic acid, not antibiotic, but clovulanic acid. So what do? On top of this, I now add not a ampicillin resistant bacteria, sensitive bacteria earlier, but now I will add here not a sensitive bacteria, but a ampicillin resistant bacteria. So I will grow my flask, I will grow my cells which are ampicillin resistant, but I will also add ampicillin into this. I will add ampicillin, and the addition of ampicillin plus ampicillin here is not going to kill my blue cells from dying because they are already ampicillin resistant. You get my argument? And then when these red cells grow and we see what happened? And we see that blue cells are growing, but these red cells are giving us red clearance zones. I now know that these cells are dying. This cell here is producing something which kills these cells. Producing something which kills these cells. And the answer is obvious, producing something which kills the cells. So either it is producing something which is toxic, which means some new antibiotic. Either it is producing a new antibiotic, I'm happy. Either it is producing a new antibiotic which is killing it, or it is somehow preventing this resistance from acting, so it is converting the ampicillin resistant to ampicillin sensitive. And the proof that it is a new antibiotic or it is a conversion can be simply done by taking the cells, overlaying it and removing, making the plate ampicillin minus. If it is ampicillin minus and the clearance zone disappears, then you know that ampicillin is required to kill it. It is ampicillin which is killing it, and what the cell is doing is aiding the ampicillin to kill cells which were earlier resistant to ampicillin. So obviously it is an ampicillin. So now this is a double screen. This is a clever screen, much cleverer than the earlier screens that we designed. This design is now ensuring that the cell is able to produce the beta lactamase inhibitor, BLIC, let me call it, beta lactamase inhibitor chemical. And so if you put this BLIC along with ampicillin and start selling, you are very rich. So this clever design was thought about by somebody 15-20 years ago who discovered that ampicillin बाजार में बहुत बिक रहा है and उसका production का technology तैयार है, तो अब हम एक ऐसी चीज निकालते हैं जो ampicillin resistant cells को भी ampicillin sensitive बना दे. And so this was an extremely good marketing opportunity. Just this splitting strategy allowed him to find out. And now the person who discovered clovulanic acid is a multimillionaire, because everywhere in all antibiotic sales, clovulanic acid has to be added. Because he discovered that these cells kill only when ampicillin is also present, but not when ampicillin is absent, which means it is not producing a chemical which kills by itself, but it produces something which makes ampicillin more effective. And the way it makes it more effective, they discovered by reverse engineering, is that it produces a chemical which is a beta lactamase inhibiting chemical, which was clovulanic acid. And now, therefore, it is added in everything. So you see, a discovery which rides piggyback on an ancient discovery of antibiotic. Right? So it takes care of the problem of antibiotic. So there are clever designs.
Sir, I have a question here.
Yes, please.
So the red cells, as in the ones producing clovulanic acid, so
Right.
those are resistant to antibiotic, as in ampicillin?
Yes and no. Yes, mostly yes. and the way we do it is very bad if they are not earlier. I mean, earlier we did not know how it was. I did not provide the details. What we do is we cover this with a sheet of film, which allows chemicals to go through, and then we actually, I mean, so those are the more complex, and then we put a layer of agar, which is a buffer zone, assuming that the whole thing will leach up and not enough antibiotic will come down. And the amount of antibiotic that we add is actually very less. So that, I mean, these are the optimization parts of any plating process. I will not get into that. You are right. These are not, I mean, obviously I'm trying to explain the concept here. The detailed experimental protocol is part of the microbiology course. Okay. So I don't want to give you I just wanted to give you the idea. The actual working protocol to take care of these minor issues is a matter of very fine tuning the whole design of experiment. And that is not an easy job. You're right. It could get killed. Because this is a soil bacteria, it may just get killed. So a good BLIC inhibitor chemical, which is producer of clavulanic acid, might actually never be discovered because it got killed. And so there are tricks to ensure that the antibiotic does not leach down, but the clavulanic acid leaches up. I mean, so the buffering layer has to be so thick that it ensures that the clavulanic acid producing bacteria do not see too much of ampicillin, and yet enough clavulanic acid goes up to actually create a clearance zone. Yeah. So now, is this clear? So
yes,
now this very sudden question, which I find it very nice. So now I have discovered, and now this is the point that I'm trying to make. I have discovered, you're familiar with the fact that cells can be introduced with plasmids which contain the antibiotic resistance gene.
Yes, sir.
ठीक है ना? तो ये बीटा लैक्टामेज प्रोड्यूस करता है, सेल ऑलरेडी है. अब मैंने एक और प्लाज्मिड लिया है. I have found another plasmid which contains beta-LIP. So beta lactamase inhibitor protein. ये भी available है. कुछ there are many proteins which can inhibit beta lactamase inhibitor protein. Need I say more? Okay, I will say more. Then of course the answer will become slightly obvious, but no, it is not very obvious. Now this presence of this plasmid makes the cell ampicillin resistant. But presence of this cell along with this BLIP, this is BLIP, and this is BL, beta lactamase. So if only this is present, then it is ampicillin resistant. If both are present, then it is ampicillin sensitive. You understand?
Yes, sir.
Hello? Yeah. Now I'm giving you this system. Design for me a screen which will pick up 10 cells out of 10 million cells, randomly, which pick up 10 cells from 10 million cells, which help extracellular export. Secretion, which helps secretion. You understand the question? Hello?
Yes, sir. Yes,
All of you? Now, the answer, of course, is obvious. So this is where I stop, and let us have 10 minutes or 5 minutes of the class, introduction class, of the next course. Or at least, or if you want me to finish here and allow you to ask questions, and maybe tomorrow on. Tomorrow, Saturday is also a class, right? Hello?
Yes, sir.
Yes, sir.
Yeah.
Tomorrow
So
will
Saturday,
work.
maybe. Yeah. So maybe Saturday we start our engineering. ये मैं यहाँ खत्म करूँ, इंजीनियरिंग शुरू करूँ 5 मिनट के लिए, या आप लोग इसमें कुछ सवाल पूछना चाहते हैं?
Sir, I have a question, but not related to this.
Okay. Related to exam, I can't answer. And grading. आप लोग एग्जाम और ग्रेडिंग का बहुत सवाल पूछते हैं, इसलिए मैं पहले से ही बोल रहा हूँ कि अभी मैं कल उसका आंसर दूंगा. आप कोई एकेडमिक डाउट है तो पूछिए.
So sir, like there was E. coli, new strain of E. coli which came in 2011 in Germany. So that was about like not because of plasmid perhaps. I was reading that in that strain like only 40 to 50% was genome was matching with earlier E. coli, but still we say that that is E. coli. So like how can we say that it is E. coli? And also like
usually what we do is we do rRNA, 18S RNA, 16S rRNA sequencing. Okay, and if the 16S rRNA is matching, and that is the very simple way to say that it is the same species. Okay, so usually we never sequence the whole genome. To if we have 16S RNA matching, we simply say, "Bhai sahab, bilkul theek hai na?" And then if we discover suddenly that the genome is very, very different, then questions arise. And since I do not know the specific problem that you are mentioning, I cannot give you a clear answer. But usually, the standard genetic method of, other methods of, it is not that before sequencing there was no way to characterize bacteria or any species. There were morphological traits, there were growth traits, there were usually how did you characterize this stuff? Earlier you used to characterize it through the microscope. So morphological characteristics, growth, shape, etc., and of course biochemical characteristics, because you could always do tests. So does it degrade sugar? Does it produce acid? Does it produce this chemical? So biochemical tests. Does it produce acetic acid? Does it grow well on glucose? Does it grow well on lactose? Does it grow well on xylose? Does it consume C5 sugars? Can it live on this? Does it produce this enzyme? So biochemical tests and morphology, how it looks in a microscope, what are the shape, size, motility, etc., were the ways to define or group bacteria, do the classification tests. Then came genetic tests, and we started looking at 16S rRNA. And now we can sequence the whole genome. So before we sequence the whole genome, usually we look at these things as indicators that they belong to a particular subgroup, a particular classification. So maybe, I do not know the question that you are raising, so I cannot give an answer, but maybe the biochemical test, the morphological test, and the 16S RNA test showed that it is E. coli. And so in a traditional sense, it is E. coli. But if it was E. coli, 99% or 95% of the genome should be matching. So if you say for 50% matching, it might have been a very interesting problem to address as to how this has happened. But remember that nowadays we are now working, especially in E. coli, with what we call the reduced genome. Have you heard of the reduced genome?
Yes, sir. Yes, sir. Minimal genome.
The minimal genome, because we have got rid of all stuff which we think are useless. But as I would tell you that this is all nonsense. We do not know, and we think that what we do not know is actually not important. Is a very old habit among scientists. कि जो चीज नहीं मालूम वो बेकार होगा. So that is a foolish and a rather very superior view of ourselves. So the reduced genome actually fortunately works. But where it does not work, we do not even know that. Because we do not have what I call the dynamic model of cellular behavior. We do have static models of cellular behavior with steady state conditions, but how the cell responds dynamically and what makes it respond is still not understood. And that is why engineers cannot model dynamics at the way they would like to. When I moved from engineering to biology for the first time, I had this very silly statement which I made to my teachers who used to laugh. And I therefore should not laugh at your statements. I said, but there are 2000 proteins, 2000 reactions. Why don't I write a set of 2000 differential equations, solve it, and I have solved the problem of E. coli. I will have a model of E. coli. And it turns out that this is the most silly statement to make, but I did not know as a student. So I made this silly statement. Now I know slightly better. So most of the information about this dynamics or the rates of change, etc., are just not known. And how it is regulated, how it is controlled, what are the signaling molecules, what is the hierarchy is also not known. So there are things about E. coli Even E. coli, which we claim to have known for 20 years, forget about mammalian, where we think that just because by sequencing the genome, we know the behavior, we do not. We know some things, obviously. We know the genes, we know the proteins, we know some amount of what I would call the steady state behavior. But the dynamics of the behavior is completely unknown. And that is what makes this subject so much more challenging and interesting. Maybe in your lifetimes, you will end up, by the time you are as old as me, you will know what is important among these. I mean, you will know much better about the dynamics.
So these things are studied in systems biology and synthetic biology, right?
Yes. Among other things, systems biology, synthetic biology are two important, but not the only subjects of interest. Most of systems biology started with, as I said, steady state work. So they did start addressing thousands of proteins instead of one, but somehow it has been taken over by flux balance analysis, and metabolic control analysis did not ever take off. So FBA and all are all steady states. And steady states have all kinds of problems. I mean, the dynamics is really the challenging part. So all FBA and all modifications of FBA, even the modifications like IFBA and combining transcriptomic data with FBA are all essentially limiting the steady state conditions or defining different steady state conditions. But how they move from one steady state to the other, that dynamics we do not understand. Synthetic biology has moved into more like toys about constructing genomic circuits which perform in certain ways which are analogous to electrical circuits, and you can do that in a very interesting way, and you can make, and if you people do the iGEM conference, I mean, I was invited in 2001 where I was the only inviter, so I was told that your students will always win the award from Asia because you are the only university, but that's 20 years ago when it was launched. Today, iGEM is highly competitive. Asia region has its own, but IIT Delhi, IIT Madras, and other places where I've gone have participated in iGEM, and iGEM provides you with these toolkits to do a little bit of synthetic biology and use your creativity. So I don't know whether your group has, or your previous group, or your next group is planning to attend the iGEM conference and the competition that goes on. It is an interesting learning exercise. What all you can do with clever genetic circuits and manipulation, and what all do you want to do, since the circuits are now available as gene groups. I mean, in E. coli, so you can just, you'll be provided with all kinds of sets of genes which do all kinds of interesting regulatory work. For example, you've done electronics. Can you design a JK flip-flop or a master-slave circuit? Can you design an AND gate or an OR gate or an AND gate or an OR gate?
And or now this gate, but not earlier things. What did you say?
हाँ, and or gate तो बहुत easy है ना. And or gate बनने लगेगा. And or gate is very easy with two plasmid systems. But थोड़ा सा और आगे सोचिए ना, मतलब ये तो हो चुका है. जो चीज हो चुका है उसको repeat मत करिए ना. That is why I said the course also is trying to emphasizing the creativity of students. I mean, and the problem with such creative talk is that it eats into the classroom. That is why I cannot do too much of this. But yes, so both synthetic biology and system biology still do not capture the essential unknowns of the cell. That is what I'm trying to tell you. And those essential unknowns will be captured over the next 20-30 years. By that time, I will be safely and properly dead. So it will be upon you guys. Okay. So let us finish this lecture. Tomorrow, let us back to engineering, no more such discussions. Let us do a few rounds of engineering classes, standard, proper, regular, conventional engineering classes. And then we will, after 3-4 lectures, we will have this quote unquote random discussion on how biology, where biology is going and what are the creative things that you can do. I think it's important, but it should not overwhelm the course.
Sir, why electrical engineering people like, I know that there are many professors even in IIT Delhi in electrical engineering who work on these the genetic circuits and etc.
Yes, so that is the whole idea, right? I mean, most people, you people have done control theory, right? You have done proportional integral derivative control, feed forward control, PID control तो आप लोगों ने किया ही होगा, and you would have done stability of circuits, Nyquist criterion and stuff like that. I mean, in your control classes. Now it turns out that what are the regulatory methods for biological control, and they have very interesting insights that they provide you. First of all, there are hierarchical controls, and they provide you very interesting insights on the ways that cell regulates homeostasis. So all cells would be like to be in homeostasis. And how does this homeostasis, how is it achieved in the face of random environmental fluctuations? Is a very interesting and instructive exercise in how a cell manages with, I mean, you don't have all these fancy electrical gadgets, and yet the cell manages under most conditions to retain homeostasis. And that is why people work on genetic circuits, because they provide a very beautiful insight and a very creative insight of how to do what I call control. Control is the real reason for studying genetic circuits, and of course also biologists are interested for understanding this. So okay, thank you very much. I think it's already 10 o'clock. We will stop this class now. We'll stop this class now, and we will discuss this next class. Okay. Thank you very much.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you.
Thank you.